Hi, in this video, I'm going to be discussing the mechanisms of action of different drugs. I'm not going to talk about all the drugs, I just picked a few drugs to talk about. But the drugs I've picked in this first video are some of the drugs that you commonly come across. And the objective of this video is to serve as a quick reference for students who are going for examinations and who might not have time to go back to the textbooks to start reading the mechanisms of actions of each of the drugs one by one. So the first one we look at are the diuretics. And we know that diuretics, we have the loop diuretics, the thiazides, the osmotic diuretics, and the potassium-sparing diuretics. So for the loop diuretics, examples are furosemide, torsemide, etaclinic acid. And these drugs act by binding to and blocking the function of the Na plus K plus 2CS importer in the thick ascending limb of the loop of Henle. The thiazides and thiazide diuretics include hydrochlorothiazide, clothalidone, indapamide, and others. And these ones act by inhibiting sodium chloride transport in the distal convoluted loop by inhibiting the Na plus Cl symport. Acetazolamide acts by inhibiting carbonic anhydrase in the proximal tubule, while the osmotic diuretics, example mannitol, isosorbide glycerin, and urea, prevent sodium and water reabsorption in the proximal tubule and descending limb of the loop of Henle. The potassium-sparing diuretics, examples of which are spironolactone, epinephrine, amiloride, and others, reduce sodium reabsorption in the collecting tubules and ducts by blocking the aldosterone receptor, that is for spironolactone and epinephrine, whereas amiloride and tramterine act by direct inhibition of sodium entry through the epithelial sodium channels in the apical membrane of the collecting tubules. For antibiotics, we have the different classes of antibiotics as we all still remember. The first class, the beta-lactams, examples of which are penicillin, cephalosporins, carbapenems, and monobactam, act by inhibiting the synthesis of bacterial peptidoglycan cell wall. Then the aminoglycosides, examples of which are gentamicin, streptomycin, kanamycin, inhibit protein synthesis by binding to the 30S ribosomal subunit. The tetracyclines, typified by tetracycline, doxycycline, minocycline, act by inhibiting protein synthesis, and they do this by binding to the 30S subunit of the bacterial ribosome. Chloramphenicol, on the other hand, bind to the 50S ribosomal subunit to inhibit bacterial protein synthesis. The macrolides, erythromycin, clarithromycin, azithromycin, also inhibit protein synthesis by binding to the 50S ribosomal subunit of bacteria. Ketolides, e.g., telithromycin, share the same mechanism of action with macrolides. Then for vancomycin, vancomycin inhibits synthesis of bacterial cell wall by binding to the D-alanine terminus of cell wall precursor units. Quinolones, on the other hand, inhibit the enzyme DNA gyrase and topoisomerase 4. For antituberculous drugs, isoniazid inhibits synthesis of mycolic acids in the mycobacterial cell wall. Rifampin inhibits DNA-dependent RNA polymerase of mycobacteria, leading to suppression of initiation of chain formation in RNA synthesis. Ethambutol, on the other hand, causes inhibition of arabinosid transferases, which are involved in cell wall synthesis. Pyrazinamide causes inhibition of mycobacterial fatty acid synthase, which is involved in mycolic acid synthesis. For the antifungals, amphotericin B, this acts by binding to ergosterol to cause formation of pores by pollines in the fungal membrane, and this will allow leakage of small molecules out of the funga. Flucytosine, this acts by inhibiting thymidylase synthase, leading to the impaired synthesis of DNA. For the azole antifungals, examples of which are fluconazole, ketoconazole, itraconazole, these ones act by inhibition of 14-alpha steroid demethylase to impair ergosterol synthesis. Then the echinocandins, example of which is capsufungin. These are competitive inhibitors of glucan synthase to disrupt beta 1,3-D glucan synthesis. Crisaofovin bind to polymerize microtubules to cause disruption of the mitotic spindle. Terbinafine act by inhibiting fungal squalene epoxidase to block ergosterol synthesis. For the antiviral agents, acyclovir is a competitive inhibitor of viral DNA polymerase, and it is also incorporated into viral DNA where it causes chain termination. Amantadine and rimantadine act by preventing viral replication through inhibition of the uncoating of viral RNA in infected cells. Oseltamivir carboxylate and zanamivir cause inhibition of influenza neuraminidase enzyme. Ribavirin causes alteration of cellular nucleoside pools and inhibition of viral messenger RNA synthesis. Then the interferons act by interacting with cellular receptors and activation of the JAK-STAT signal transduction pathway, leading to inhibition of viral penetration, translation, transcription, protein processing, maturation, release, increased expression of major histocompatibility antigens. For the anticancer agents, the alkylating agents, examples of which are cyclophosphamide, busulfan, chlorambucil, ifosfamide, they alkylate DNA by forming reactive transition complexes like carbonium ions. The platinum coordinating complexes, examples of which are cisplatin, carboplatin, oxaliplatin, form equated compounds that react with nucleophilic sites on DNA and proteins to cause formation of interstrand and intrastrand cross-links, and also inhibition of DNA replication and transcription, leading to breaks and miscoding. The folic acid analogs, examples of which are methotrexate and pemetrexate, inhibit the enzymes dihydrofolate reductase and thymidylate synthase. 5-fluorouracil inhibits thymidylate synthase, thereby preventing synthesis of thymidylate. The Vinca alkaloids, vincristine, vinplastine, act by binding to beta-tubulin to prevent its polymerization with alpha-tubulin into microtubules. The taxanes, paclitaxel and docetaxel, act by binding to beta-tubulin of microtubules to prevent its disassembly, thereby causing mitotic arrest. The epipodophylotoxins, etoposide and teniposide, bind to topoisomerase II DNA complex to prevent resealing of DNA strand breaks, thereby leading to DNA damage. The camptothecin analogs, topotecan and irinotecan, bind to and stabilize the topoisomerase I cleavage complex, thereby leading to DNA damage. The anthracycline antibiotics, doxorubicin, daunorubicin, epirubicin, and idarubicin, act through several mechanisms. 1. By binding to DNA and preventing its transcription. 2. By generation of superoxide anion radicals, which can produce hydrogen peroxide and hydroxyl radicals, which damage DNA. And 3. By inhibition of topoisomerase II. Hydroxyurea, on the other hand, acts by inhibiting ribonucleoside diphosphate reductase. L-asparaginase acts by catalyzing the circulating asparagine to aspartic acid and ammonia, thereby depriving the malignant cells of the asparagine required for protein synthesis, leading to cell death. Tamoxifen is a competitive inhibitor of estrogen binding at the estrogen receptor. Fulvestrant acts by binding to estrogen receptor to inhibit its dimerization and increase its degradation. Anastrozole, aminoglutetimide, exemestane, all are inhibitors of the enzyme aromatase, thereby preventing the peripheral conversion of androgens to estrogen. Ganirelix, abarelix, cetrorelix are competitive inhibitors of gonadotropin-releasing hormone by binding to the GnRH receptors in the pituitary. Gonadorelin, goserelin, histrelin, leuprolide, nafarelin mimic the action of GnRH at the GnRH receptor. Flutamide, bicalutamide, nilutamide are selective antagonists at the androgen receptor. Trastuzumab is directed against the human epidermal growth factor receptor 2, thereby inducing an immune-mediated response that causes internalization and recycling of HER2, resulting in downregulation of the HER2 receptor. Zoledronate acts by inhibiting osteoclast proliferation and induces osteoclast apoptotic cell death. Extramustin binds to beta-tubulin and microtubule-associated proteins to cause disassembly of the microtubule and antimitotic effects. Cetuximab is a monoclonal antibody directed against the extracellular domain of the EGFR. Bevacizumab inhibits VEGF interaction with VEGFR1 and 2. Inhibits VEGF interaction with VEGF receptors 1 and 2. And drugs used for the treatment of asthma. These are beta adenoreceptor agonists, examples of which are salmeterol, albuterol, levabuterol, metoprolol, tabutalin, and others. These ones cause stimulation of beta-2 adrenergic receptors in the bronchial smooth muscles, which in turn will lead to activation of GS adenylyl cyclase cyclic AMP pathway with consequent reduction of smooth muscle tone. Glucocorticoids, e.g., beclomethasone, dipropionate, triamcinolone, acetonide, and others, act by 1. Modulating cytokine and chemokine production, 2. Inhibition of eicosanoid synthesis, 3. Marked inhibition of accumulation of leukocytes in the lungs, 4. Decreased vascular permeability. Then the leukotriene receptor antagonists, zafilukast, pranlukast, and montelukast, are competitive antagonists of leukotriene receptors. They are selective, high-affinity competitive antagonists with inhibition of cis-LT1-induced bronchial smooth muscle contraction. Then LS1, that is leukotriene synthesis inhibitors. The example there is Xilutone, and that is currently the only drug in that class. Is a selective inhibitor of 5-lipoxygenase, thereby inhibiting the formation of all 5-lipoxygenase products. Omalizumab is an anti-immunoglobulin E antibody which inhibits binding of immunoglobulin E to mast cells. It also inhibits the synthesis of immunoglobulin E by lymphocytes. Lastly, the methylxanthines, examples of which are theophylline, theobromine, caffeine, inhibit cyclic nucleotide phosphodiesterases, thereby preventing the breakdown of cyclic AMP and cyclic GMP. The anticholinergics, tiotropium and ipratropium, are competitive inhibitors of acetylcholine at the muscarinic cholineceptor. Then cromolyn sodium and nedochromin sodium act by 1. Inhibition of mediator release from bronchial mast cells, suppression of the activating effects of chemotactic peptides on human neutrophils, eosinophils, and monocytes, inhibition of parasympathetic and cough reflexes, and lastly, inhibition of leukocyte trafficking in asthmatic airways. Thank you. We'll take a different set of drugs in the next presentation. Thank you.